Welcome everybody that's here and here on Zoom. Again, my name is Kevin Lang. I've been working with the research office to organize this seminar series, which we're calling the RIDE seminar series, to capture the research, innovation, discovery, and education aspects in the college. So today, we're starting off with our first RIDE research seminar with two internal speakers. We have Dr. Ferenc Toth, who's going to be starting us off today from Vet Clinical Sciences Department, and then followed up by Dr. Bruce Walcheck in the Veterinary Biomedical Sciences Department. So I'm not going to take up too much time speaking, and so I'm going to hand it off to Ferenc and have him tell you about his current research.
Good afternoon, everyone. Thank you very much for coming. I was told to say a couple of words about myself. Unfortunately, my direct advisor or mentor, I should call, at this point on main campus, giving nearly the identical talk, which I'm giving right now here. So the audience is clearly split. It looks like she took the majority with her because she's clearly better, but she is also would have been the one who introduced me. So my name is Ferenc Toth. I'm originally from Hungary, hence the atrocious accent you are experiencing right now. And I did most of my training at the University of Tennessee, including my large animal surgery residency, as well as my PhD. And then I got tired of the hot weather and came up here enjoying the cold. But look around, you just can't move north enough, apparently. So anyways, my presentation today is titled Update on the Development of Large Animal Models of Orthopedic Diseases. And this was an ambitious title before I realized I only have 20 minutes to talk. So the diseases should be taken into a singular here. And I will talk primarily from one condition. I don't know that one was. And this is not working. Sorry. My first slide. Okay, so what I'm going to talk primarily about is going to be osteochondrosis dissecans. And I will start with a brief introduction. Then we're going to talk about the goat model of the disease, which we try to develop. And we try to use this goat model to investigate two aspects of osteochondrosis. One of them was using MRI to identify generally the cartilage lesions that we are inducing. And then we also try to look at the cartilage canals, which are very important over the course of the disease. This one was done in goats and other species as well. That's the reason why it is a separate heading. And then I will speak a little bit how we drifted to mini pigs from goats, and then how we drifted from our original site of the distal femoral condyle to the trochlea. I was also planning to talk about Lechalve-Perthes disease, but I will not have the time, so I apologize for that. Starting with the introduction to osteochondrosis, osteochondrosis dissecans is a developmental orthopedic disease. This is defined as a focal failure of the endochondral ossification. Animals and human beings are affected at predilection sites, and these predilection sites are shared across species. That was one of our motivations to start to pursue this project, largely for its translational aspect. It results in a formation of osteocartilaginous flaps or fragments, and if these are left untreated, then they tend to progress to osteoarthritis, increasing the importance of the disease. As far as how the disease really look, Ryan, it's a silly question. Does it have a laser pointer? I take it as a no. Laser pointer? Sorry.
For a sec.
Yes. That's a laser pointer?
Oh, you just have to kind of like.
Oh boy, okay, that's not gonna work. That's good, thank you, all right?
I can turn into a laser.
That's quite all right. So what the lesions are, then concentrate on the arrows, please. So the lesions on the top left, that is a human ankle, and you see a characteristic OC lesions affecting the trochlea in the ankle. And you can see at to the right of it, two images which are obtained with horses. And horses also have these lesions potentially serving as a natural model. Underneath it, you can see the most common predilection site affected in humans, and that is the medial femoral condyle on its axial aspect, again marked with an arrow. You can see it in a horizontal and a coronal plane image. And again, you can see a corresponding lesion in horses. Getting back a little bit to the pathogenesis of the disease, which is necessary to understand the rest of the presentation. I'm going to be the laser pointer. So this is how a developing joint looks like. This is an ossified region, and this is the articular epiphyseal cartilage complex. And that, due to its thickness, is at least partly supplied to vessels directly running through it. And during the development of osteochondrosis, what happens is that for some reason, which is not clearly understood why, there are several speculations, the vascular supply is interrupted to a circumscribed area of the cartilage, which undergoes necrosis. This is the earliest lesion, what one can see with osteochondrosis. In the, I don't know how to put it, like earlier times, it was only able to recognize using histology, and it was unavailable for any kind of medical imaging techniques. I would like to believe that our work on the medical imaging side now made these lesions apparent, and that's what I'm going to try to show you, how our MRI development resulted in our ability to be able to diagnose the disease at this earliest stage. If you looked at histology, this is what you would be seeing, what you can see here, a very distinct area where cartilage necrosis is present, which is embedded into normal cartilage. Since it's only affecting the cartilage, that's the reason why you couldn't see it on your radiographs. As the disease progresses, as the animal ages, the ossification front progresses towards the articular cartilage, and it kind of embeds or starts to surround this lesion. And since the ossification front is visible on radiology, at this stage you can see the lesions. This is usually the time when it is getting diagnosed. It is called osteochondrosis manifesta accordingly at this time. On gross pathology, you can see these stair-tap lesions over there. Histology, this is how it looks. The good thing about osteochondrosis is that it can go two ways from here. In some cases, it can undergo, in the severe or the unfortunate situation, it becomes clinically apparent if this lesion, if this area which has the lesion is exposed to trauma because of the insufficient support this necrotic island can provide to the overlying cartilage, it cracks through, and this results in the clinical disease which is called osteochondrosis dissecans. However, if this lesion remains protected from excessive trauma, then the ossification front slowly surrounds this lesion, and it eventually with time resolves, and you are away scot-free. You're gonna be healthy and not limp for the rest of your life. So the existing gap, was a brief introduction which I thought is necessary for everybody to understand really where our work is and what we are trying to do. But the existing gaps what we are trying to address is why the pathogenesis of osteochondrosis is well understood in animals. That's what I showed, for the pure reason that you can euthanize animals without any symptoms, and you can do these sentinel studies and identify this preclinical disease. This is not possible in humans. Accordingly, in people, the pathogenesis of osteochondrosis was largely not well understood. There is absence, and accordingly, there is absence of evidence-based guidelines of how to treat this disease in humans. Therefore, we thought that an animal model would provide us an opportunity to develop a minimally invasive imaging methods which are able to identify these earliest lesions, ideally a non-invasive way, so you can use it in children. And it would also facilitate the development of various treatment methodologies as well as diagnostic modalities. Accordingly, the overarching aims of our work is necessarily threefold. One is to develop an animal model of human osteochondrosis, and then we also try to image these early changes in the extracellular matrix which are associated with the condition using MRI to identify a lesion at their earliest stage, and also image the cartilage canals because we know that their interruption is what leads to the condition to develop. So if we can image those cartilage canals in their healthy state, then we should be able to make conclusions we should be able to identify the disease based on changes in them. So going to the goat model of osteochondrosis, our aim in this part of the study, and this is where our research started, was purely to induce osteochondrosis-like lesions in young goats by interrupting the vascular supply to the articular epiphyseal cartilage complex. And for this, we initially conducted preliminary studies just to describe the vascular supply to the distal femoral epiphyseal cartilage, which was largely unknown at that time. And based on this, we designed a surgical procedure in which we operated in, again, 4 to 5 day old goats. Altogether, we enrolled 8 goats into this part of the study. The red number indicates that 3 of these goats underwent a more invasive procedure. 5 of them had just the vessels transected, which we were able to identify in our perfusion studies. and they are marked with the dashed lines on the images, one on the side of the lateral trochlea, one on the axial side of the medial femoral condyle. And in those 3 peaks, we also used the scalpel and created a horizontal incision to transect those vessels which are traversing the ossification front. So after we completed this procedure, these goats were euthanized 1, 2, 3, 4, 5, 6, and 9 weeks after the surgery. And since we wanted to get some success, at least initially, we went straight for a 9.4 Tesla magnet, which has a greatest resolution, better signal to noise ratio, although it is not available for human imaging, but we just wanted to see what we can get. And these are our initial results. So at 2 weeks postoperatively, we were able to, doesn't project very well. I'm going to step one up, actually 2. So if you look very closely, there is an area over here which has like a, I don't know, like a fainter coloring to it. If you look under the microscope, it is much more obvious. It is as early as 2 weeks after the interruption. But what is more exciting here, that we start to pick up these lesions on our MRI, more apparently on the adiabatic T1 row sequence than on the other one. But I don't think you have to use too much of your imagination to notice over there the increased signal. As the lesion progresses, so does the signal intensity. By 5 weeks, it is really hard to miss the lesion in the articular epiphyseal cartilage complex. And by this time, it is also more pronounced on histology. You can clearly see the island of necrotic cartilage, which is trying to delay the progression of the ossification front. Here it is using safranin O staining. At 9 weeks, the lesion is starting to definitely impinge on the ossification front. So in this case, we would probably call it as an osteochondrosis manifesta lesion. And you can see both of our MRI sequences very clearly. So it was all fine so far. We were very excited. And then we ended up with this. Unfortunately, the goat which was carried out to, it's not so unfortunate, that's a fact of life. The goat which was carried out to 10 weeks, we started to notice that these animals are healing their lesions. So instead of the course of going through trauma and developing clinically apparent disease, these animals tended to recover. So taken together, what we found so far is that surgical interruption of vessels supplying a portion of the articular epiphyseal cartilage complex resulted in ischemic cartilage necrosis, which is consistent with the earliest form of osteochondrosis, osteochondrosis latens. And these lesions with time progressed to osteochondrosis manifesta. That is, they delayed the progression of the ossification front. However, they did not develop into clinically apparent disease. More importantly, we found that there are 2 sequences which we were able to use at 9.4 Tesla to clearly identify the lesions at the earliest OC latent stage. So that was a positive sign, which we noticed that maybe this thing has legs, and we should be able to develop something with which we are able to identify these lesions well before they were able to be identified before this work. So the problems we were facing is that the surgically induced lesions failed to progress toward apparent OC. So we need to somehow improve our model to trigger clinical disease. And then also imaging was performed ex vivo at 9.4 Tesla, which is not something you can do in humans. So we have to show feasibility at clinical field strengths with clinically available sequences. So the second portion of our study, our aim was to expose now these goats after the surgery to some form of exercise and hope that the trauma which is associated with that results in the development of clinical disease. We got 4 goats, the same age as previously, and did the same procedure as I described. But after that, 3 weeks after the surgery was done, by the time we know there were OC latens or OC manifesta lesions present, we exposed these goats to some exercise. And this is how exercising goats is like. I would be a lot better on a treadmill if somebody sat me while I'm doing it. But it hasn't been instituted in my gym yet. Nobody is, bringing food around me when I'm doing my stuff. Now, granted, I'm also not euthanized after it, so I guess I take that. So this is the goat exercise, and this is what we try to do to impose some trauma on them. And we also did some in vivo MRI imaging at 3 Tesla, and we shifted from our research sequences to T2 mapping based on advice of our MRI scientist, and then euthanized the goats and evaluated our findings using histology. So this is the first set of images that we obtained at 3 weeks out because we wanted to see before the goat started exercise if there is a lesion where we operated on. And we found that 4 out, one case is not shown, but 3 out of 4 cases had a successful lesion induction, which was clearly apparent or somewhat apparent on MRI. As you can see, the increased signal intensity marked with the red in the areas. So this is a failed one. You can see the cartilage is entirely normal, proximal to the incision, whereas in this case, there is clearly a difference in the relaxation time in the operated area. Going out, oh, I could have done this. Going out to 6 weeks, you can see that the one which was failed, there is not much change. Whether those animals which had successful surgery, the lesion persists. And then at the time of euthanasia, these findings were confirmed histologically. You can see that one of the 4 goats operated on, 402, developed a crack extending through the lesion. So this is by definition meets osteochondrosis dissecans. However, in the other 2 goats which had successful surgery, nothing but an osteochondrosis manifesta lesion persisted. So we came to the conclusion that surgical interruption of the vessel supply combined with exercise resulted in OCD development in 1 of 4 goats. More importantly, we found that T2-weighted imaging at 3 Tesla was able to identify the earliest lesions, that is osteochondrosis latens, also known as ischemic necrosis of the AE complex, as early as 3 weeks postoperatively. But we are facing remaining problems, that is the low yield of the procedure, which was 1 of 4 goats. And I think the ladies who were doing the exercise with the goats also got into much better shape because of the rather intensive nature of the training. So this one made us a little bit to think that what is going on? Why is that if humans have these lesions and they exercise, they develop clinical disease, whereas in our goats there was only 1 of the 3 which progressed on, and that made us think about that maybe it is somehow related to the vascular supply. Reason being that vascular failure is what leads to the disease. It also contributes to the healing. Maybe there is a difference. So what we decided to do is to take a look at how the vascular supply to the epiphyseal cartilage looks like, if it can be visualized, and if it can be visualized, how does it compare among species? So this was a long and technical study, which was, how I should say, it was our brainchild, but the true mind power behind it was workers of MRI physicists at the CMRR. So I'm certainly not going to go into details what kind of sequence development has taken place because I have a very limited understanding of it. But how we did it, we started with ex vivo imaging of small samples. Then we went over and tried to image a whole joint, still ex vivo. Then we tried to do in vivo imaging at a clinically relevant field tank. And ultimately, they came up with a method which is called qualitative susceptibility mapping, which relies on susceptibility information from instead of the direct phase information from MRI. And based on that, they were able to generate exceptionally nice 3-dimensional reconstructions of the vascular supply, which enabled us to compare species. And what we found was quite striking. It seemed to be our bad luck to select the goats for our initial species, because if you take a look at humans and pigs, I would like to believe that their vascular supply is very apparently similar. If you take a look at it, you can see that the vascular supply coming from the sides, either axially or abaxially from the joint, and the vessels are running horizontally, that is parallel with the joint surface. You can see both people and pigs have the same thing. One of the many traits, I guess, we are sharing with pigs. This is unlike with goats, because if you take a look at the goats, it looks like that the vasculature much more like a cobweb. There are lots of anastomosis among those vessels. Much less of those are apparent both in humans and pigs. And then there is also a vast majority of the vessels in goats, as they age, start to come across from the ossification front. So when you try to interrupt vessels axially or abaxially, you are not going to have the same results as you get in humans and pigs. So based on this recognition, we shifted and we decided, so what we found is that the vascular supply to the complex can be visualized using susceptibility weighted imaging. By special processing method, we were able to eliminate some artifacts which got in the way of our 3D reconstruction, and we were able to identify these marked differences among animal species which are free of the disease versus those are affected. And based on this, we decided that we need to change and from goats pivot towards pigs. I have about 5 more minutes, and we started a little late. So we went to the miniature pigs and start to do the rest of our work there. And based on the similarities in the vascular supply, we decided that we can go and grab 28 to 30 day old piglets and operate on their femoral condyles. Trying to increase our yields, we do 2 kinds of surgeries. So we remove the 5 by 5 millimeter segment of perichondrium, either from the axial aspect of the right medial femoral condyle and or the left abaxial aspect of the medial. So in the right limb, we remove the segment from the abaxial aspect. In the left limb, we did the surgery on the, I'm mixing it up, right limb axial, left limb abaxial portion of the perichondrium was removed. And then these pigs underwent MRI imaging 3, 4, and 9 weeks postoperatively before euthanized and evaluated histologically. What our findings were here is somewhat meeting our expectations, I would say. So we found that out of the 4 pigs we worked on, in 3 animals, we were able to induce OC manifesta or OC latens lesions in those cases when the surgery was done in the abaxial region, and this has not happened after the axial surgery. How it exactly looks on MRI using our now clinically available sequences, I think this is probably the best example from a pig. We were able to see it as early as 3 weeks. You can see this increased intensity involving the epiphyseal cartilage, even at the early stage of the disease. And as the disease progresses, it's becoming more and more apparent. And at the time of euthanasia, histologically, the lesion was clearly identified. The lesions in the other pigs remained somewhat smaller. So at this point, again, we had to think a little bit what's going on. Why is that the abaxial surgeries worked, the axial surgeries didn't work? And we couldn't come up with a better explanation that during the surgical procedure, it was me who was doing the surgeries. I was facing tremendous difficulties to access the intercondylar area. And although I'd like to believe that I did a good job and we indeed resected the given segment of the perichondrium, truthfully, it was probably not accomplished, and this get into the way. But that doesn't take away from the fact that the surgery from the abaxial side was also rather difficult, and it also didn't yield exactly the outcome we were hoping for. So we again went back to the drawing board. And although the majority of human lesions affect the femoral condyle, and that's the reason we were also targeting it, we started to realize that maybe it is better to just shift a little bit and approach the trochleae, the femoral trochleae, which have a much better surgical access. They are also involved in the disease in humans, and try to work out some of the kinks over there. And this takes us to the last part of this presentation, which is induced lesions of the femoral trochlea. And this is actually a fairly recent project. In fact, the hopefully a publication, a manuscript is undergoing review right now, I should say. Again, in this study, we have relied on the fact that OCD affects both the condyles and the trochlea in people. The femoral trochlea in pigs are free of naturally occurring disease. So if we operate on the trochlea, we can be sure that the lesions, what we see, are due to our intervention and not naturally occurring disease. It has a structured conserved vascular supply, which we found in our vascular imaging, which again lends it well for the surgery, and it offers excellent surgical outlet. So these 3 factors were those which strongly contributed our new approach. Our aim was to determine the relationship over here between the severity of vascular injury to the femoral trochlea and the formation of the progression of lesions of OC in these pigs. So we, in addition to try to shift to a new location, we also try to see if by increasing the lesion size, we are more able to induce clinically apparent disease. We used 10 pigs in this study, and we performed surgery in the lateral trochlear ridge, trying to interrupt the vascular supply. This first image showed a very clear vascular supply. Hopefully, you see my mouse? No, it's just not working. All right, so this is the vascular supply, which is very well defined. So when you are in surgery, you are not really fishing for anything. You can see what you are operating on. And we do two kinds of surgeries. We either remove the entirety of that vessel, including the perichondrium, trying to affect the entire lateral trochlear ridge, or we just went and removed the distal half of that vessel, trying to affect that portion of the trochlea. After that, we did MRI at 1, 6, and 12 weeks postoperatively and euthanized the pigs and analyzed them histologically at 12 weeks. Our results are probably best shown in these images. So this is a pig in which we have conducted resection of the entirety of the lateral trochlear ridge vascular supply. You can see the findings 2, 6, and 12 weeks postoperatively. The top set of images are cartilage maps, which the arrows very clearly show at 6 weeks, at 12 weeks, the very defined lesions. There is an increased intensity also at 2 weeks, but it is ill-defined, so you cannot measure the size of the lesion. Beneath, you can also see alternative MRI sequences showing these findings and finally histology. And this is in contrast to those pigs which only underwent surgery involving the distal aspect of the trochlea, which developed tiny lesions. Now, this you might think it's common sense that a smaller surgery results in a smaller lesion, but it was very much debated across, like the orthopedic surgery field and as high level as it can go, such as AAOS. So over here, we were able to show that the size of the initial vascular injury determines the size of the developing lesion and its course towards resolution or progression towards extensive preclinical lesions of OCD. And we also found that the induced trochlear lesions lend themselves for evaluation of the effects of various forms of biomechanical injury, which are better tailored just than a treadmill exercise. So we are developing a machine with which you can drop a certain weight on this induced lesion to see if we can promote their progression to clinical disease. And I'm sorry for going a little over. Thank you for your attention. Any questions?
Do we have any questions for parents? I have a quick question. Is there any, what is the average age in human that this orthopedic disease generally develops? And is the age of animal maybe linked to why you don't see, so much disease in your models?
So this is only develops while there is articular epiphyseal cartilage present. In case of a human, this is about the age of 14 to 16. And clinical, this is usually seen 9 to 16, between 9, the most frequently it's seen between the age of 9 and 16. We have done some studies on human cadaveric specimens, and we were able to describe the earliest lesions, which I refer to OC latens, as young as 2-year-olds, but they are just simply not ambulating and running around enough to develop into clinical disease. What we try to do with our model is capture an approximately similar age as well a 6 to 8-year-old children is. So that's when we induce the lesions and try to see how their quicker development in piglets parallel that one which is seen in humans.
I was curious with the unique features that you saw in the goat.
Yes?
Did you, it's interesting from the perspective of evolutionary biology and the evolution of goats.
Those are big words, yeah, go ahead.
Yeah, so did you all talk a little bit about that? And are there features that are interesting from that perspective that are worth tracking down?
I don't know, I think we are just a little more simple-minded than that, and we just thought that these are ruminants, and they probably have a different general developmental. I mean, I think, please don't get me wrong. I'm not dismissing your question. It is very valid, but it is just simply beyond our level of, I'm certainly not a geneticist. I don't know much about the evolutionary biology. I think it's a very valid concept. We went to the easiest route that, well, if I think about ruminants, very rarely have OCD. So unfortunately, we didn't even have the opportunity to take a look at cows. It would be interesting to see a juvenile calf, how their vascular supply looks like. But based on the fact that they don't develop OCD, I would bet my hat on it that it is going to probably look like a goat. And maybe it has a very clear evolutionary background. I don't know. Yes, ma'am.
Hi. Thanks, Ferenc.
You are welcome.
Very interesting. I'm just wondering, kind of related, these are, have you looked at animals in the wild? Basically, this idea of the amount of activity and moving around and how constrained animals are. Otherwise,
I have not, but I think there are studies looking at wild boars, and I am going to go out on a limb here, and I think they did not describe the lesion in wild boars. So I am not sure what is the reasoning behind it. It's a very valid question. I guess they get eaten by the wolves by the time they develop the lesions, but yeah, I don't know. I am not about. Actually, I take it back. There is a very recent publication which shows it, I think, in a tiger, an OCD lesion, but in an entirely different form. So that tiger had some sepsis, and there were vascular thrombosis in those vessels, and that resulted in necrosis, and which ultimately developed in the same clinical manifestation, but it had a different background. So the vascular failure originated from sepsis-related thrombosis. But other than that, I don't believe there are many studies looking at it. I'm sorry.
I think in the interest of time, we'll move on. Thank you, Ferenc. Let's
You're
thank
welcome.
Ferenc.
Thank you. Sorry, didn't mean to be so needy with the laser. It's still on. Thank you.
Yep. Good. Great. All right. So I am speaker number 2, and I was told when I was invited to give this seminar that a person from my lab could introduce me, but I thought that would be a very dangerous platform for them. So I opted to introduce myself, and so my name is Bruce Walcheck. I'm a professor in the Department of VBS. Got my PhD at Montana State University, did my postdoc at this company called Boehringer Ingelheim in Ridgeville, Connecticut, and then came to Minnesota and have been here at the university ever since. So this is the ride seminar, and I will take you on a ride where we will learn about natural killer cells. So this is a, do I have to point at this? Oops. Oh, I see what I'm doing now. Okay, got it. So we have myeloid cells and lymphoid cells that make up our leukocytes, and natural killer cells are a type of a lymphocyte. You're probably more familiar with T cells and B cells. They're the majority of the lymphocytes. NK cells are probably, what, 20–25% of your lymphocytes. But they're a bit of a different type of lymphocyte population, where T cells and B cells are antigen specific. A T cell expresses a T cell receptor, a B cell makes an antibody, where an NK cell uses a whole array of receptors on their cell surface to interrogate cells in the body. And so they're referred to as being part of the innate immune system instead of the adaptive immune system. So in this slide, just an illustration of some of the various receptors that these NK cells, natural killer cells, express. We're not going to get into them except for one, but they really fall into 2 categories. Either these receptors are inhibitory or they're activating. So an NK cell is floating around in your body. It's interrogating the cells in your body to determine whether they're healthy or they're abnormal. Have they been infected by a virus? Are they a transformed cell or altered in some other way? So when they interact with a normal cell, the ligands on the normal cells interact with these inhibitory receptors, and that turns the NK cell off, keeps it in a resting stage. But if the cell it interacts with is an altered cell, it has ligands on its surface that interact with these activating receptors, and that will turn the NK cell on. And when it gets activated, what it does is basically just release a bunch of cytolytic factors that will interact with the cell that it has bound to, and it will cause that cell to undergo programmed cell death and kill itself. And NK cells also release a lot of pro-inflammatory factors too that have a broader influence on the immune system. And so I'm going to be talking about antibodies today. So to refresh your memory about antibodies, I have this illustration here. Really just the two things to remember about an antibody for this seminar is it has this Fab region. This is the region that binds to antigen. And it has this FC region, and this is the region of the antibody that can interact with complement or be recognized by FC receptors. I was going to mention here that of these activating receptors, there's this one called CD16A. This is really its most potent activating receptor. And what it is it's an FC receptor. It binds to the FC region of IgG antibodies. So CD16A is an IgG FC receptor. It's also referred to as FC. Gamma stands for it binding to IgG antibodies. R is for receptor, and 3 is because there's 3 family members in this family, and it's the third one. And I'll get into that in a second. But so we have CD16A binds to the FC portion of IgG antibodies. If these antibodies bind to a target cell, like a tumor cell, CD16A on the NK cell will interact with that FC region. That will send signals into the NK cell, and it becomes activated, releases its cytolytic factors and cytokines and such. So let's see, this process is called antibody-dependent cell-mediated cytotoxicity, or ADCC. And this is a process that's being manipulated in terms of trying to enhance the ability of NK cells to kill tumor cells. And so one point of leverage to manipulate this process is to alter the receptor itself, CD16A. But CD16A has a couple types of regulatory processes that reduce its function to control it because it's such a potent activating receptor. And one of these mechanisms is it's just a low affinity receptor compared to other family members like CD64, which is a very high affinity receptor. I'll get into that in a minute. Another way of regulating it is cut by a protease called ADAM17, and that quickly just downregulates its cell surface expression. You get rid of CD16A, it's not going to be interacting with antibodies on target cells anymore. So this is a very quick way of regulating the function of CD16A. But if we want natural killer cells to kill tumor cells better, these are processes that are sort of a disadvantage and points of leverage that we're trying to manipulate so these NK cells work better. So with this protease that cuts CD16A, we had determined exactly where it cuts. It cuts in this region very proximal to the cell membrane. We have mutated that region, and we can prevent that cleavage from occurring. We just call this non-cleavable CD16A. We described this a number of years ago, and since then, this has been engineered to be expressed in NK cells, and the function of these NK cells have been examined, and they've been shown to mediate ADCC better. That's reported in this publication here, and I really wasn't going to get into that today. We have patented that engineering of CD16A. It has been licensed to this company called Fate Therapeutics. They've engineered it into their NK cells and T cells, and these are being currently tested in phase 1 clinical trials. We were interested in version 2 of this receptor. And so all this work has been done in collaboration with Jimmy Wu in our department. And so let's see, so CD16A is part of a larger family. There's 6 members in this family. And CD64 is the only high affinity receptor that binds to IgG. It binds with an affinity 50 to 100 times higher than CD16A does. But otherwise is very similar, binds the same isotypes of IgG as CD16A does. And so we were interested in making this fusion receptor where we have CD16A, here's the two extracellular regions, part of this slide has disappeared, but transmembrane and cytoplasmic region here. We have CD64, it has one additional extracellular domain, contributes to it being a higher affinity receptor. We took the extracellular region 64 and fused it to the transmembrane and tail of CD16. So we have the high affinity portion of 64 and the potent signaling component of CD16A. And CD64 is not cleaved by atom 17, so we don't have to worry about that regulatory process either. As a quick aside, most of this work is related to enhancing human NK cells. CD16A, but I should say we are also interested in sort of reverse translating what we've learned in the human to apply this to the dog as well. So can we use some of these immunotherapies we're trying to develop in the human and develop them in the dog as well? CD16A and 64 have not been characterized in the dog. So with the help of Jimmy Woo, we clone them, we express them, we made antibodies to them, and we have characterized those receptors in the dog in terms of which dog leukocyte populations express them. And that's described in this publication here. I'm not going to get into that, but we find that CD16A really seems to be the best marker for NK cells in the dog as well. And so we have some other projects going on related to that. But I was going to talk about CD6416 today. And so we want to express this in NK cells. But NK cells are a difficult cell to genetically engineer because when you alter an NK cell likes to kill a cell that has been altered. So you alter an NK cell and they just like to then kill themselves. So what we've done instead is we've gone back to this induced pluripotent stem cell. This can be differentiated into a hematopoietic cell and then eventually differentiated into an NK cell. And the advantage of this is these iPSCs are very genetically manipulatable. So we can put our genes of interest into them and just differentiate them into NK cells. This technology was developed here at the University of Minnesota by this Dan Kaufman. He has left, but we have brought this in to our labs. And so Jimmy Woo and I are doing this in our labs. And so basically just shown here is we make these mature NK cells. We call them iNKs because they came from iPSCs. But basically when we make an NK cell, they have the markers of an NK cell. And when we stain them for CD64, they stain at very high levels, picking up our construct that we put into them. If we just make iNKs but don't genetically engineer them, they do not stain for CD64 because they do not express our construct. Okay, so a problem with making these cell-based immunotherapies is that they tend to be very antigen specific. They will target just one tumor antigen. The problem with that is many types of tumors are very heterogeneous in the antigens and ligands that they express. So one type of receptor that has been generated that probably most people have heard of, it's called CAR, chimeric antigen receptor. This is generated and typically expressed in a T cell. These are FDA approved to treat a couple different types of cancer as standard therapy in humans. Typically lymphoma is a common one that's treated. They're basically just a construct of a piece of an antibody that will recognize your tumor antigen, a transmembrane region in the signaling domain. So when it interacts with an antigen, sends a signal into the T cell gets activated and it kills the cancer cell. The problem with this is it recognizes one antigen. So when you engineer T cells to attack tumor cells, it works really well in killing the tumor cells that have that antigen. But typically what can happen is there will be tumor cells that don't have that antigen. They will then start to grow and they will cause relapse of this tumor because they're now resistant to this therapy. And so a problem with this therapy is it's only targeting one antigen. And so we're trying to move forward with therapies that can target more than one antigen. And we think that's an advantage of our approach because our targeting element is an antibody. And there are a lot of different FDA-approved antibodies out there that target a lot of different tumor antigens. And there's a lot more in the pipeline that are being generated. So we don't have to generate that tumor antigen targeting element. That's already out there and available. Our receptor just binds to that antibody. So we express our receptor in the NK cell, and we don't have to change receptors. We can just change antibodies to target new antigens. And so you could treat your tumor cell with a particular antibody to target a certain tumor antigen. Come in with our cell, with our receptors, it will bind and then kill that tumor antigen. Another thing we can do with our cells is because this receptor 6416 is so high affinity, we can just dock or couple the antibodies right to these cells. And that has particular advantages as well. And for instance, we can just dock or place on these cells different types of antibodies to target different tumor antigens. We just call this antibody arming of these cells. And so I was just going to briefly talk about utilizing these cells in our lab. This is just an assay that allows us to measure the coupling of these antibodies to our NK cells. For the sake of time, I'll just skip that, but it works very efficiently. So I was going to talk briefly about two assays we use to examine the function of these cells. One is an in vitro assay and one's an in vivo assay. This is our in vitro assay. It's called an Leukocyte live imaging assay. What we do is we take our tumor cell of interest, we incorporate a gene into it that when expressed in our tumor cells will fluoresce a particular color, like it will fluoresce green or red. And when these cells are eliminated by our treatment, we just lose green and red cells, and you can just measure that in the instrument. Or you can put a dye in there that detects apoptosis, and you can measure earlier cell death by that process as well. So basically what we do is we take our tumor cell of interest, our NK cells of interest, and whatever else we want to put in here, put them into a well of a 96-well plate. This goes into our Leukocyte instrument. This instrument is housed in an incubator, so these cells are able to be cultured under physiological conditions for an extended period of time. We can read these fluorescent signals at various intervals, seconds, minutes, hours, out to several days. So that's one advantage of the assay. And so what happens is, let's see, in this assay, we just have two different examples of this assay. We have our tumor cell, it's called a SCOV3. It's an ovarian cancer cell line. It fluoresces a particular color like green or red. Then we just add our iNK cells that have been armed with an antibody or not armed with an antibody. So our readout is over here. Time is hours. We're going out 48 hours and basically percent of living tumor cells in our assay. Our SCOV cells are the blue line. They increase in number over time because they're just growing normally. If we put our iNK cells in expressing 6416, but not armed with an antibody, they will start to kill these tumor cells because they have other receptors that they can use to kill those tumor cells. But if we arm them with an antibody, they kill these tumor cells much better. This 16:1 represents 16 NKs to one tumor cell. If we have 8 NK cells to one tumor cell, we see the same thing, just the level of killing is a little less. And so here we just have I and Ks. They don't have our receptor. We attempt to couple antibodies to them, but they don't have our receptor, so the antibodies don't stick to these cells. And we just find that if we've attempted to arm them with antibodies or not, they really just kill the same, and they kill much less efficiently than our armed cells. And just quantifying the data over various repeated experiments here. Okay, last thing I wanted to get into is just our in vivo experiment for looking at this. So here, again, we manipulate our NK cells. Here we express luciferase in them. So if we add a reagent like luciferin, it will metabolize that. It'll cause these cells to luminesce, and we can visualize that in the mouse in an instrument we use. So we inject our tumor cells. Again, here we're injecting SCOV cells. We put them into the peritoneal cavity because this is the main site of metastasis for ovarian cancer cells. And then we can inject luciferin in these cells and visualize the location and size of our tumor, and then just measure that in this instrument called an IVIS instrument, which is available on the Minneapolis campus. So this was our treatment scheme here. We inject tumor cells in at day -4. That's the red arrow. These black arrows is when we put them in the instrument to do our bioluminescence. So basically doing that every week. We do it at day -2 to look at the initial implantation of our tumor and to group our mice so they all have similarly sized tumors. We inject our NK cells, that's the blue arrow, and we just inject them several times for multi-dosing. And I'm not going to get into this gamma guard right now for the sake of time. And here for this experiment, we had 3 groups of mice. Either they received a tumor plus our INK cells not armed with an antibody, or our INK cells armed with an antibody. These are cryopreserved. We arm our cells with these antibodies. We wash that antibody away, and we put our cells into liquid nitrogen. This is a particular advantage in the clinic where you can take the NK cells out of a freezer versus having to grow them fresh and put them into a patient. So we wanted to add this to our assay to kind of increase the stringency of our assay. So all these cells have been cryopreserved, thawed, and then injected into the mice. And then just real quickly, this is probably hard to see, especially in the back, but these are our different groups of mice. There's 6 mice in each group. These blue, the various colored dots are the tumor that are luminescing. These are the days that we're doing our analysis. So you can see upon initial injection, you can visualize the tumor. In our tumor alone mice, the tumor gets larger, and eventually you have to sacrifice some of our mice, so we start to lose some of our mice over time. If these mice are treated with INK cells not armed with an antibody, there is some reduction of our tumor, but the tumor continues to grow. If these mice are treated with INK cells armed with our antibody, the tumor actually gets smaller over time. And that's quantified here. Blue bar is tumor getting larger over time. Red bar is mice that got INK cells not armed with an antibody. Tumor is reduced but continues to grow. And mice that got tumor plus INKs armed with their antibody, the tumor actually shrinks over time. Had another version of this experiment, but I'll skip that for the sake of time and just get to the conclusions. So here we generated the 6416, incorporates the high affinity version of 64 and the potent signaling region of CD16A. Because of its high affinity state, we can arm our INK cells with antibody. These cells induce potent tumor cell killing in vitro and in vivo. And in the future, we hope that these cells may provide an off-the-shelf, that means it comes from the freezer into the patient, like taking a drug off the shelf and giving it to a patient. Strategy for It's multi-tumor antigen targeting to overcome tumor relapse due to antigen escape. And that's our story so far, individuals in the lab. The bulk of this particular work for this story was done by Kristen Snyder, who's a DVM PhD student, and some of it was done by Kate Dixon, who's a postdoc in the lab, and she has another story related to another type of cancer that I didn't talk about today. And we collaborate very closely with Jeff Miller's group over on the Minneapolis campus. All right, and I'll end at that.
Questions?
Thank you. Fascinating talk. Bruce, I might have just missed it, but when you look at the in vitro experiment in the in vitro incubation, you said one was 1 to 16, the other one was 1 to 8 ratio NK cells to tumor cells. And I'm not sure if I misunderstood or not, but it appeared that the 116 did better than the 1.8, assuming that when you had a lower ratio of natural killer cells to tumors, they seemingly killed better. It, for some simple-minded person like me, sounds counterintuitive. What do you have any explanation for that?
So when your ratio is 16 to 1, 16 NKs to 1 tumor cell, they do kill better. Yeah, yeah. Right, yeah. Yeah. Right. The opposite. And I went through that pretty quick. So yeah.
Okay, Bruce. So we were talking just before the seminar started about CAR T cells and their problems with the FDA. So on with medications or with anything, there are off-target effects, and you mentioned that the NK cells can release a lot of things that can affect the immune system. So do you have a way of turning this off if you have to? Is there something you can give to, say, induce some of those inhibitory receptors on NK cell to kind of shut this down if you had to?
Yeah, we think that's a particular advantage of the NK cells as well. These CAR T cells, again, not to, I guess, crap on that procedure. It works really well, but it has a lot of issues. Antigen escape, this cytokine release syndrome. When you attack lymphoma cells, it may kill those lymphoma cells, but eventually they kill your B cells as well, and that's a lifelong problem. So there's a lot of issues with that potentially can be resolved with NK cells. First of all, NK cells are a shorter lived cell, so you're not going to have this lifelong persistence of them. It's an allogeneic therapy where the T cells, the CAR T cells, those T cells come from the patient. So the patient's got to have enough of those cells to even engineer them and put them back. Ours is allogeneic. They're coming from a donor. Well, actually coming from an iPSC, so it's an unlimited source of these cells that you can continue to give to that patient. And NK cells, if you want to turn them off, especially by our approach, you just take the antibody away and they have no more targeting mechanism. Yeah.
So I'm going to ask a question really quick. What will the NK cell do if the target of the antibody is not another human cell? Like, what if it's a virus or something else?
What would the antibody do?
The antibody, well, the armed NK cell.
Okay. Well, if the antibody is not to an antigen that came from that virus.
No, well, let's assume. Can you use it to clear virus out of?
Oh yeah. Actually, that is something that is being looked at for like HIV to target cells that have those particular antigens on their surface. There's clearly antibodies available to those antigens. Can you use those antibodies to target NK cells to those virally infected cells? Yeah.
Thanks, Bruce. That was so clear. 2 questions real quick. On the protease cleavage site, is that only in these NK cells, or is that, are there other targets of ADAM17?
Right, ADAM17 is a very interesting protease, something we study a lot in the lab. My graduate student who just defended a couple of weeks ago gave a seminar on that protease. It cuts a lot of different receptors. CD16 is one of its best characterized substrates, but it does cut other receptors on NK cells. ADAM17 is really expressed on every cell in the body, and it's got substrates on those cells. They're not CD16, they're different. So it does a lot of different things. It's something we're interested in targeting as well, The risk is it cuts a lot of different things and what might be the off-target effects of that. But
And the other one was the armed NK cell. So you mentioned when they like to kill each other if they don't look like the self. So is that a pretty short turnover protein? Do we have to worry about off-target effects, or does it just pretty much have other molecules kill itself?
yeah, that is an issue with these antibodies, therapeutic antibodies, is they're not really to a tumor-specific antigen. They're to a tumor-associated antigen. These antigens can be found on normal cells, so you are going to have the risk of collateral damage from this. This is just a risk you face with any type of immunotherapy. There's going to be collateral damage. So that is something you have to take into account and try to control. But at least with our approach, you can just take the antibody away. The half-life of an antibody isn't that long, and then it's no longer present.
Very exciting work, Bruce. So the mouse model is in a control experiment where you use the SCID IO2 receptor knockout conditions. And this will be, I imagine, individualized medicines, right? If you're going to bring it to treatments for human, how do you envision this going into the clinics? Do you have to, under what conditions would you have to do to the patients, and how often do you have to infuse these into humans?
Well, the advantage of this approach is it is not as individualized as CAR T-cell therapy, FDA-approved CAR T-cell therapy, because those T-cells come from that patient. Our NK cells just come from this induced pluripotent stem cell. This is, again, something that Fate Therapeutics specializes in. So, and this is an advantage of NK cells I won't get into, but they can be administered to really any individual because they don't have a T-cell receptor on them that's going to attack the patient's own cells. They don't have that capability. That is a problem with a T-cell. So you can give these NK cells to a much broader range of patients. So that's a particular advantage. Again, they can be cryopreserved. That's a big advantage. But there are disadvantages. These patients, you have to essentially knock down their immune system to accept these cells better. You do probably have to give them in multi-doses to get them at a level and a persistence that sticks around long enough to really treat that tumor cell. So these are certainly disadvantages that people are attempting to work around and improve. Certainly nothing is perfect at this point, but these are things that can be addressed.
Awesome. Let's thank Bruce one last time. Okay, thank you folks for coming and everybody on Zoom. If you can, just two things real quick. One is if you could please take this survey if you've been to this or any of our events. So if you are, not with it, you just point your phone at this thing and then it'll tell you what to do. It's really quick, just 2 questions. And then also join us next month. We're having our first invited speaker, Dr. Anne Belser from Auburn University. And then after that in May, we're having again 2 of our own, Dr. Noel Noyes and Dr. Steve Friedenberg, talk about their current research. So thank you again, everybody, for coming.